everybody, this is our last video for this unit, and it's all about glycogen, making glycogen and breaking down glycogen. For some quick terminology here, when we're talking about making glycogen, it's called glycogenesis. Here we are storing glucose. We're just stringing a bunch of glucose molecules together, hooking them onto a protein. There's a protein called glycogen, and that's on the next slide, so I won't write it down here. But we're just storing any excess glucose that we have, and it ends up kind of branched out like a little tree. So it's kind of like that. Now, when we need to get those glucoses back, so we want to break down our glycogen, we do glycogenolysis. Glycogenolysis, we're just taking those glucoses back off of our little glycogen tree, and we do that at the end. So we're not going to just cut a little piece off of the middle of our glycogen molecule. We're going to be snipping off the ends here. Now, you do glycogenesis when you have a lot of glucose available. When you're making a lot of energy, you've been in, you've eaten, you're in a fed state, so that's why there's a buffet going on behind here. You just ate the buffet, you've absorbed a lot of glucose, you've done a lot of glycolysis, you're making a lot of energy, so you have this excess amount of energy and glucose and all the byproducts of those things, and then you don't really need to be doing glycolysis and Krebs cycle and all that anymore because you have enough energy. So you can start pushing your glucose more into the glycogenesis pathway, which is just another shunt coming off of glycolysis again. So again, starting with glycolysis. Glycogenolysis, when you're breaking down your glycogen, this is when you don't have enough glucose. You need more energy and you need it quick. You want to break down your stored glycogen, take away the glucose molecules off of that glycogen so that you can get that glucose back into the blood and then into the cells. Now, there are things that regulate when we push our glucose into the glycogenesis shunt, or if we're going to go the opposite direction and do glycogenolysis. Now, in our fed state, so after we've eaten, we have typically some hyperglycemia, so you have a lot of glucose in your blood that you just absorbed from your meal. Insulin is the hormone that is released at that time. You have a lot of ATP, a lot of citrate for the Krebs cycle because you're doing glycolysis going into pyruvate and acetyl-CoA and then joining up with citrate and running the Krebs cycle. You have all these things in high concentration, and all of those things encourage glycogenesis. And when we look at the pathway, I'll show you how to kind of push it that way. Now in the fasting state, so you haven't eaten for a few hours or so, you start to get hypoglycemic, so you don't have a lot of glucose in your blood. Glucagon is the hormone you release in that situation. You have low ATP inside the cells because you're not doing glycolysis. You are getting energy from fats. We'll talk about that a little bit later, but it's just lower. It's not as intense as when you're in the fed state. Your citrate is a little bit down. Again, same thing. It's not gone. You're still doing Krebs cycle starting from fats, but it's just a bit lower because it's a slower process. All of these things push you towards glycogenolysis to break down the glycogen. Now, epinephrine, it's not part of the fasting state. Epinephrine is the fight or flight reaction, your adrenaline. So this could happen at any time, fed or not fed. But this also pushes you to glycogenolysis. And we'll see how and why. It has a lot to do with the enzymes, always the enzymes. Whatever the relative concentrations of enzymes are, that will, determine which way a reaction is going to go. Okay, so let's just look at glycogen and its structure. Glycogen is made mostly in your liver, so your hepatocytes. Make glycogen. Also your skeletal muscles can make glycogen. Just a bunch of glucoses all strung together, stuck to a protein called glycogen, or glycogenin, not glycogen. Glycogenin is the protein. And it's just keeping your extra glucose in a safe place to be used later if you need it. As we, were to break that down, we would do that if we were in, hypoglycemic sort of situation. We would be breaking it down from the end. So we can grow the molecule in all these little branches, and then we can break it down at the end. Okay? Okay, now here is the pathway for glycogenesis. So this is our glycogenesis pathway. And I want you to notice one thing, or a bunch of things on here actually. Look for glucose. Glucose in this unit, because we're talking a lot about glucose and glucose metabolism and all that kind of stuff. When you're presented with a metabolic pathway, look for the things that look familiar so you have a starting point to interpret what's going on in the rest of this diagram that looks pretty complicated. But if you find your familiar points, then you can just do it one little step at a time and work your way around this particular pathway. So there's glucose. Glucose is not intimidating. We all know glucose. There it is. So that's our glucose right there, our sugar. Then we look up here and we see glucose 6-phosphate. Well, we know that too. That's the beginning of glycolysis and the pentose phosphate pathway. So there we go. We could, then end up in the Krebs cycle maybe. So, pyruvate, acetyl-CoA, Krebs cycle, we could go that way. So we know this part of this chart already. These are the enzymes involved in turning glucose into glucose 6-phosphate, and we know it's reversible. We can, go this way, we can go this way. Now, if we have our situations here, so hyperglycemia, insulin present, ATP, lots of ATP, lots of citrate, we don't really need to do this. We've done enough of that. We're going to instead go this way. And we're going to change our glucose 6-phosphate into glucose 1-phosphate. And now we are in the glycogenesis portion. So we are growing the glycogen molecule. Or I should say not growing, but beginning the glycogen molecule. And as we go over this way, then we are kind of breaking, or continuing to grow, I should say, we're not breaking it down yet, we're growing the glycogen molecule. And then as we go down this way, then we're returning right to the beginning. If we're returning to the beginning, then that means we're breaking it down. We're going back to glucose 1-phosphate. We're breaking away the glucose from the glycogen. So over here we're doing glycogenolysis. So it goes full circle. We start glycogenolysis with glucose 1-phosphate. We grow our glycogen molecule, we grow it, and now we're breaking it down and we're back to here. So we would be doing this part of the cycle under these circumstances, and we'd be doing this part of it over here under these circumstances here. Now let's just look at the rest of it. This is the main points there, but if you just look over here. We got a lot of stuff going on, a lot of enzymes. So glycogen synthase, that pretty much tells you it's synthesizing glycogen. It's an enzyme that's going to be synthesizing glycogen. We don't have to worry so much about over here. These are side pathways that we're not really caring about. But what we are caring about is glycogenin. That's our protein that we're going to link our glucose molecules onto using a glycogen primer, which is just another molecule that is going to help us with our enzyme, glycogen synthase. We have our, glucose byproduct here, a glucose 1-phosphate going into this uridine diphosphate glucose. So essentially a glucose molecule ready to be stuck to glycogenin. And now we've started to form our glycogen molecule. We haven't branched it yet. I'm just getting ahead of myself. We're not branching it. We're just starting to form the glycogen molecule, hooking this uridine diphosphate glucose onto the glycogenin using the glycogen primer, and the enzyme glycogen synthase is facilitating that. Okay, so here, this is talking about how we link which carbon. So we're linking the number 1 carbon of one glucose molecule to the number 4 carbon of another glucose molecule. Now we get our branching enzyme. This is where we start kind of going like that. Instead of one long chain, we have little branches. And this is referring to, here's the 1 to 4 link again, so the number 1 carbon of one glucose, the number 4 carbon of another. Here's the number 1 carbon of one glucose and the number 6 carbon of another glucose. So we start forming these 2 different sorts of bonds, just where the glucose molecules are bounded together. And we continue growing that molecule. We just keep growing that molecule, getting it bigger and bigger and bigger. So we just have more and more branches because of the branching enzyme. And here we have this glycogen phosphorylase. You see PI here, that's phosphate, getting kind of incorporated into it or being used to continue to grow this molecule. And this glycogen phosphorylase is stimulated by, all these situations here. These situations that cause you to do glycogenesis. And so is glycogen synthase also. So the more glycogen synthase you have, the more glycogen phosphorylase you have, the more you're kind of making your glycogen molecule. So beginning it here, building it over here. Now, if we were to then, once we built our molecule, if we were to then get into some of these situations, then we would start to go this way, where we have a debranching enzyme, or glucan transferase. That's another enzyme that will help the debranching enzyme to start kind of skipping off little pieces of glucose, breaking it down, doing glycogenolysis. And you notice that these things, these are doing 2 things. One, we're getting a free glucose from our debranching enzyme, but we're also taking some of these glucoses that have been transformed. Remember, they got transformed into this uridine diphosphate glucose. But now we're putting them back into the form of glucose 1-phosphate. Glucose 1-phosphate, that was what we started with. So we've gone back to our starting point. And this is reversible. We can go back to glucose 6-phosphate from here. And you think, here we are, we're hypoglycemic, got glucagon going on, we have low ATP, that's kind of like the big key there, low ATP. What do we want to do? We want to do glycolysis and the Krebs cycle. So we broke down our glycogen, we converted those single glucose molecules back into glucose 6-phosphate, and now, boom, we get to do glycolysis. Now, the next slide has the same picture on it, but it doesn't have all the writing that I did, so do that, leave that there so we can still see all these notes. And this is just reiterating or saying what I just said, that we're starting with glucose. Find your starting point. Find the thing that's really familiar. There's glucose 6-phosphate. That's also familiar. And then, okay, now we're in a different pathway. This is a shunt. We know there's glycolysis. That's our main pathway. This is the shunt. This is a different way to go. And we can just follow around the glycolysis. We have choices of where we can go from the glucose 6-phosphate. Doing glycogenesis, not glycolysis, glycogenesis up here. We could go to glycolysis in the Krebs cycle. We could go to the pentose phosphate pathway. So we could, do a variety of things with this one little molecule of glucose 6-phosphate. And then this point here is that it's all reversible. We can then take our glycogen that we took all this time making, and we can bring it back down again. And, go back to our original choice and end up doing a different way instead. Okay. So, glycogenesis, starting with glucose 1-phosphate, we saw that on our chart. When we have lots of glucose, so hyperglycemia, lots of ATP, lots of citrate, also insulin. Glycogenolysis, it ends with glucose 1-phosphate, so that's the full circle part. Glucagon, adrenaline, hypoglycemia, low ATP. So all the things that we saw here before, those are the things that stimulate glycogenesis or stimulate glycogenolysis. Okay, so here we go. Glycogenesis after fasting. So remember, fasting just means you haven't eaten for a few hours, or it could be a longer term fast, like an official you're on fast kind of thing. But even just a couple hours, your body's going to start to run out of glucose, because glucose is a really quickly burned sort of molecule. It's just used for energy really, really fast, so it can get used up pretty fast. So when you're in the fasting state, when you haven't eaten for a little while, you start using lipids to make ATP instead of glycolysis. Now you use lipids a lot of the time. You use lipids pretty much any time that you don't need energy really, really quick. If you can wait for the energy, it's more efficient to use lipids as your energy source. They make more energy per carbon than glucose does, but it just takes longer. There's more of a process to it. So if you need fast energy, you're using glucose. If you are doing slow energy, then you can use lipids instead. Now, the process for breaking down lipids into acetyl-CoA that you can use in the Krebs cycle is called beta oxidation, and that's what takes a while. But this whole beta oxidation inhibits the enzyme pyruvate dehydrogenase. Now, pyruvate dehydrogenase should sound familiar to you at this point. You've heard it a bunch of times already. It's when we're talking about glycolysis, the end of glycolysis, we get pyruvate. We need to convert the pyruvate into acetyl-CoA to go into the Krebs cycle. And we use pyruvate dehydrogenase as the enzyme that does that. So you need pyruvate dehydrogenase to take pyruvate that you have and turn it into acetyl-CoA. Now, when beta oxidation is inhibiting pyruvate dehydrogenase, that means that any pyruvate that you have is not going to be able to get turned into acetyl-CoA. And we know that if it can't get turned into acetyl-CoA, it becomes lactic acid. Okay, so lactic acid is not a good thing, but we have a way to deal with it. The way that we take care of that lactic acid is we take that lactic acid to the hepatocytes in the liver, and they do glycogenesis on it because of all these reasons that we had here. So pyruvate from just your regular cells, not able to do the whole process not able to turn into acetyl-CoA, because when the lactic acid goes to the liver, it gets turned into glycogen. Okay, so glycogenolysis. Remember, glycogenolysis can be stimulated by glucagon, and it can be stimulated by epinephrine, which is a form of adrenaline. Okay, so epinephrine, adrenaline, this is the specific adrenaline that we're talking about is epinephrine. And you release glucagon when you're hypoglycemic. Now, glucagon and epinephrine are going to bind to receptors on your hepatocytes and on your skeletal muscle cells, and they activate a second messenger system, which is not familiar to you from A&P. A second messenger system is essentially where you have the cell membrane. There is a receptor on the cell membrane. There's something called a G protein. It's sort of stuck on the inside of that receptor. And then here comes your, let's just use glucagon. There's glucagon molecule, it binds to this receptor that activates the G protein, and when that G protein is activated, it in turn activates a bunch of different chemical reactions. Now, the chemical reactions activated by this particular G protein and hormone combination goes in this order. First, CAMP. CAMP is something that tends to stimulate cells. Whatever it is that the cell is going to do, CAMP makes it do it more. It increases the activity of a cell. And in this case, it will do it by activating another enzyme called protein kinase A. And what protein kinase A does is it activates glycogen phosphorylase, which starts sending us down this side of our pathway. Remember, over here, we are starting our glycogen molecule. With glycogen synthase then starting to link the glucose molecules together, the branching enzymes start causing them to turn into like a little tree shape. So over here we are growing our glycogen molecule. And then as we pass by the glycogen phosphorylase area, then we start getting into glycogenolysis. So by increasing this enzyme, we are pushing this reaction down into the bottom end of the entire circuit. So we're going back towards glucose 1-phosphate. Now insulin would just do the exact opposite of that. It would kind of stop everything here. It would, or slow down this process anyway, or this side of the process, sort of allowing everything to kind of stall here in the growing phase, getting bigger and bigger and bigger, as we don't allow it to continue on and complete the circle. Okay, so that is it for Unit 1, and that is all that will be on our exam one.